Thank you, Alexis. Welcome everyone to today's session, which Alexis said it's computer-aided design, modeling, and simulation. Personally, I find this topic very, very interesting. It's very close to my heart. It's something I'm working on currently. So yeah, today we have 4 talks in this session. And the first talk will be given by Payton Thomas. The name of the presentation is A Comparison of Weighted Stochastic Simulation Methods. Payton is a second-year undergraduate student in biomedical engineering and applied mathematics at the University of Utah. He is interested in the behavior of biological networks and the nonlinear dynamics of cellular systems. So Payton, please, whenever you're ready.
All right, thank you, Martin. Let me screen share here. All right, can you see my screen? Just anyone? Okay, yeah. Right, so as I was introduced, I'm Payton Thomas from the University of Utah, and I'd like to present the work of myself along with my collaborator, Mohammad Ahmadi, at the University of South Florida today concerning weighted stochastic simulation methods. So I'd like to begin with a bit of a motivation for what we're going to see today. So, of course, the traditional method to simulating networks of chemical reactions would be to write down a system of differential equations from our chemical kinetics laws and then simulate or maybe even analytically calculate the behavior of that system of differential equations. And this gives you a deterministic model. So a given set of initial conditions will always lead to the same system trajectory. But unfortunately, this technique really breaks down when we talk about low molecule counts. And that's because it's dependent on sort of approximating the concentration or the molecule count for each chemical species to like a differentiable variable, which they aren't, because the number of molecules is, of course, always going to be a discrete natural number. So when we have, 5 molecules, that's maybe a little further from a continuum than when we have millions and millions and millions. So immediately this suggests, maybe naively, that we could perhaps simulate a chemical reaction network where we have this issue of low molecule counts with molecular dynamics. So give every particle an initial position, an initial momentum, and then using kinematics, calculate their trajectories and see how the system would evolve. And the reason I say that is naive is because that there's a lot of computational costs associated with such a method. Of course, once you have any reasonable number of molecules floating around, it's quite difficult to simulate their position and momentum at all times. So that really leads us to the sort of state-of-the-art way to do this, and what we're going to be talking about today, which is this idea of stochastic chemical kinetics, which is this method where we describe the chemical system as evolving according to some probability law. And then probabilistically, we can produce many, many, many different system trajectories and then talk about the probability of certain things happening or not happening. And this is really in contrast to the differential equations methods because this is non-deterministic, right? So the same initial conditions will lead to many different trajectories, and we have to produce many of those to get an idea of system behavior. So this need for a stochastic simulation method for chemical reaction networks is addressed by the SSA, or the stochastic simulation algorithm. So this is an algorithm that will choose a randomly distributed, exponentially distributed interreaction time, so time to next reaction, and then choose a random reaction according to how large its reaction rate is relative to the reaction rate of the other reactions in your system. And sort of one by one, produce the reactions and the times in this manner until you have a trajectory for your system. And you could look at many of these trajectories and approximate the probability of some event happening. So that could be, for example, the probability of some chemical species reaching a certain concentration or falling to a certain concentration, something like that. Unfortunately, this SSA really just produces these realistic trajectories for the system. And if you were, for example, trying to probe the probability of a very rare event, you could have trouble with that because it might take many, many runs of the SSA to see that event happen even once. So if an event has a probability of 10 to the -7 of happening, you would expect to need 10 to the 7 runs in order to see it occur even once, and seeing it happen once is not a good estimate for its probability by a long shot. So it can be quite computationally inefficient to try to simulate rare events, which can be an issue in biology, because of course in biological chemical reaction networks, we have rare events. We have rare epigenetic events and rare mutation events that can lead to things that are very interesting to us, like cancerous phenotypes, premature cell apoptosis, things like that. So one approach to sort of trying to probe these rare probability events with greater efficiency is this technique called importance sampling. It's called a variance reduction technique. And what we do in importance sampling is we increase the likelihood of the event we're interested in happening in some precise way, and then are able to sort of account for that change because of that precision and the sort of specific way we did that. So for sort of a first pass on importance sampling, the way we like to think about this is by imagining a dartboard. And suppose you're really terrible at darts. When you throw a dart at this dartboard, it's a uniform random variable where that thing's going to land. And say you wanted to know the probability that you, being terrible at darts, hit the bullseye. Well, if you have this little bullseye on this dartboard, you can imagine you'd have to throw many darts to get any to land in there. And we have that same problem of simulating rare events. But what we could imagine doing is maybe we increase the area of that bullseye tenfold. And then we throw darts, we see how many get into the target area, but then instead of using that number, we divide it by 10, because 1/10 of those presumably would have made it in if it were the original small bullseye. And then we can hopefully converge to the probability of you getting that bullseye faster. Now, the way we execute this mathematically is we think of the expected or mean value of some random variable x under the distribution f as, of course, the integral over the sample space of each element of the sample space times the distribution f at that point. So we can imagine in the integral, and this is just this first line of math here, dividing then subsequently multiplying by some arbitrary non-zero distribution function or density function g, which we'll call the importance density. And we can see that this integral is the expected value under g instead of f of x times f of x over g of x, which we'll refer to as a weight or omega of x. Now, so that leaves us this: the expected value of x under f is the expected value of x omega x over g. And then we could find the sample mean of this g. So we could find x omega x many, many times where x is distributed according to g, and then add those all up and divide by how many we produced, and hopefully that will converge to the same number. And the real trick to this technique is just coming up with a g that will make that sample mean at the bottom of the page there converge to the true probability faster than if we had just produced x's according to the distribution f. And, you know, we'll see that really finding this importance density, finding that G, or one that works well, is really the big issue here. And we'll see that through all of these algorithms. That's really what we're grappling with. Maybe I didn't say it explicitly. So the core idea of all of the algorithms that we'll be discussing today is this idea of importance sampling to simulate rare events. And the implementation is a little bit more elaborate because we're applying it to chemical systems. There's a little bit of work that needs to be done to quite make it work. But this is at its core, the idea. So to study these different algorithms and sort of examine their behavior, we're going to work on this 6-reaction feudal cycle motif. So it's a series of chemical reactions. You can see on the top of the page there with chemical constants given. And initial concentrations given. And we will be probing the probability of the rare event that species 5 gets to 40 molecules within 100 time units. Now this feudal cycle motif here is present in many places in biology, so it's a good example for us to study. For example, this sort of crops up in GTPA cycles, MAP kinase cascades, and glucose mobilization models. And really, just for intuition, this is two species, species 2 and species 5, being enzymatically interconverted back and forth. And there's a symmetry in the initial conditions and the reaction constants, so that the system will, with high probability, stay right around where we're starting it here. So with species 2 and species 5, both balanced at 50. So it's quite unlikely that we'll see large deviations from that, such as species 5 getting all the way down to 40 molecules. So the first algorithm we'd like to discuss is the weighted stochastic simulation algorithm, or the WSSA, which was developed by Kuwahara et al. In 2008. So this is the first algorithm that we're aware of that implies important sampling techniques to rare biochemical events. And the way this works is it applies a weighting to each reaction in the system. So it's going to change or multiply or divide its rate constant by some weight, which the user specifies. And the hope is that you can sort of identify which reactions lead to the state of interest and which ones do not, and weight them up and down respectively, and hopefully see more rapid convergence to the probability of a rare event as you generate runs on this thing. Now, unfortunately, we found that this technique is very sensitive to weighting. And to illustrate that, let's sort of apply it to our feudal cycle here. And the way we'll apply that is by generating some delta between 0 and 1. So making reaction rates lower. And we will lower the reaction rate of this reaction here, reaction 3, which produces species 5, and we will increase the reaction rate of reaction 6, which produces species 2. So hopefully we'll prefer 2 and not prefer 5, and 5 can fall down to that 40, the event we were interested in probing. And we do this to just introduce one biasing parameter to simplify analysis here. And we see this curve when we estimate probability as we vary that biasing parameter for 100 runs. Now this event has a probability of about 4%. So 100 runs is I think a pretty reasonable goal. We'll see it happen a few times with the SSA, but hopefully this will converge quicker. And what we see is that for a narrow band of values around maybe 0.6, we see really good convergence, rapid, solid convergence, but we see interesting behaviors. Now, the right side of this graph is maybe a little less interesting because, of course, when delta is greater than 1, this is reciprocal weighting. We're weighting this the wrong way, right? Not the way that we think would lead to more rapid convergence. And it's also important to note that at a biasing of 1 on the x-axis there, that corresponds to the behavior of just the SSA with no weighting whatsoever. But we see that as we go towards the left here, there's this rapid drop off, and then the algorithm begins to sort of estimate a probability of 0 every time. And it looks like this is a narrow range of biasing values here, but I'll remind you that we had delta and 1 over delta. So a 10-fold difference in the weighting parameters would correspond to about 0.3 down here. So really, if you wanted to go a 10-fold or 15-fold or et cetera, anything larger than that difference in the biasing parameters for these two reactions, we're going to estimate 0. And of course, this is with this single biasing parameter. So you can imagine that identifying that 0.6, which is very non-obvious, is challenging. But as we want to come up with a biasing parameter for every single reaction in the network, the complexity scales very quickly. Now, it's interesting that we see when we have aggressive weighting, this falling off to 0. And our hypothesis for why that's happening, to explain that, we'll return back to the dartboard analogy. So you can imagine when we weight these reactions very aggressively, we see every single path running just, in our case, that example 6 or that reaction 6 over and over again to reach the error state. And almost every path leads to the error state, but they take it using only a small sample of the total sample space of paths that could reach you to the error state. So in the dartboard example, you can imagine scaling up only a small portion of the bullseye by a very, very large number. And then when you throw darts and get them in and then scale them down accordingly, well, you're going to almost estimate the probability of just this portion of the bullseye, right? And the probability of reaching the error state taking only that one path is quite low. And that's why we see an almost 0 probability here with the estimate, because we're converging the probability of reaching the error state through a very specific path. And there is no method presented by Kuwahara et al. for determining these biasing parameters. So without that, it's difficult to use the algorithm to any real effect. The second algorithm we'd like to discuss is the state-dependent weighted stochastic simulation algorithm, or the SWSSA. This was developed by Rowe et al. In 2010. So in this algorithm, they hypothesize that maybe the struggles that the WSSA has are because As the system evolves in time, the propensity or the reaction rate for each reaction is going to vary because the chemical species vary in concentration. And of course, the rate depends on that. So maybe it's a poor idea to try to use static weights as we did in the WSSA for these events that are going to change probability naturally as the system evolves. So in the SWSSA, a maximum amount of biasing is specified. And when the reaction is quite unlikely, according to traditional chemical kinetics, we apply that maximum weighting. And when it is quite likely, according to traditional chemical kinetics, we don't really apply any weighting at all. And the hope is that this will make it less sensitive to those user-specified biasing parameters. So an example of how this biasing works is given in this curve here. So we see when the relative propensity of the reaction is quite low, we apply an aggressive biasing parameter. And when the reaction is quite likely to happen, regardless, we stop biasing it. And as we generate trajectories on this algorithm, I think we see a slightly increased robustness to user-specified parameters. We certainly don't see this falling off to 0 at extreme values, although we do see the kind of erratic behavior we saw with reciprocal weighting before. But I'll point out that through this range here where we see decent estimation, so sort of 1.5 up to maybe, I don't know, 2.5 on the x-axis here, the relative error is quite high. I mean, we see all the way up to 50% relative error, even through this range of what I would consider to be good values. And now, of course, you can reduce that by increasing the number of runs, but in doing so, you sort of defeat the purpose of the weighting, returning to just a number of runs that, well, the SSA would have done decently as well. So the same issue really here is that, you know, I mean, if we had chosen exactly 2, it looks like we could have gotten a good value, but we still see a bit of a robustness issue, and we still see this high sort of variability relative to just the WSSA. So this guides us to, nice. This takes us to our third and final algorithm that we'd like to discuss, which is the guided weighted stochastic simulation algorithm, the guided WSSA. Now, the approach of this algorithm is to, oh, and I apologize, this was developed by Colin Gillespie et al. In 2019. So the goal of this algorithm is to eliminate the weaknesses of the WSSA and the SWSSA by taking no user input whatsoever. Now, the way the algorithm achieves this is by looking at the state you're currently in and calculating using some statistical techniques, some multivariate statistical techniques, the expected number of times the number of times you expect to see each reaction in the system fire, given that you reach the state of interest right when the simulation ends. And the hope is that by conditioning that we reach the state of interest every time, but right when the simulation ends, we'll sort of barely tip the system up into the error state. We won't see this just extreme weighting, which leads to this poor estimation in the other 2 algorithms. And of course, from calculating the expected number of times each reaction fires, you can get what you think the reaction rate should be right now. So we can derive a rate from this expected reaction count. And of course, this takes no user input whatsoever. So the only input the user would give would be the system to be simulated. Now, the first weakness we really see with this method is that the technique they use, they use a normal approximation to a Poisson distribution, has negative support. So it throws up negative number of times the reaction should fire. Okay, so if a reaction should, according to the statistics, be undone a certain number of times to reach the state of interest at the end of the simulation, we'll see a negative number there, and that leads to a negative reaction rate, which leads to negative probabilities, which we of course have no idea of, no way to simulate. And we were luckily able to get a hold of the code used for the examples in this paper. And we saw that in the 3 examples in the paper, 3 different methods were used to resolve this issue of negative probabilities. So 3 different methods in 3 examples, none of which are mathematically motivated as far as we know, and none of which are presented in the version of the algorithm that was actually published. So looking at those 3 methods, we found 2 of the 3 work in general, and 1 of the 3 works on the specific example it was applied to. And between those 2 that work in general, we saw virtually no difference in performance. So we'll sort of proceed without making a distinction between those 2. But it's important to note that the algorithm as it exists it can't really be used without a little bit of extra information. Now, when we plug in one of those negative expectation methods to make sure we have positive probabilities again, and then simulate, we can see a curve like this. So the few things to notice here on slide 18. So we see quite slow convergence with the guided WSSA relative to the WSSA with an ideal biasing parameter. Just about as fast as just the SSA. So immediately this sort of undercuts the promise of important sampling to converge to this probability faster, because we see the guided WSSA taking a similar number of runs to achieve a similar relative error to the SSA. Of course, this curve also illustrates really how strong the WSSA, that first algorithm presented, is, given that you have that ideal biasing setup. So we of course saw that algorithm can perform worse than any of these with poor biasing. Now, this issue here of the large number of runs needed for the guided WSSA to achieve the same error as the WSSA with an ideal biasing parameter is exacerbated by the computational complexity of the method. So the guided WSSA algorithm requires at every step a matrix inversion. This is quite slow compared to the relatively straightforward math of the other two methods. So we see, you know, a 2 or 3-fold times increase in simulation time. This is for 10 to the 5, 10,000 runs here when we use the guided WSSA. So those runs, the same number of runs, is going to take quite a lot longer. A bit of a curious corollary of this figure here is that the WSSA, despite requiring more math than the SSA, runs faster. And we hypothesize that that's because more of the simulation runs are terminating prior to the time 100, the final simulation time, because they're reaching the state of interest. So then we actually end up with fewer simulation steps and less time simulating, despite each simulation step requiring more calculation. So some conclusions to take away from this. Firstly, the performance of the WSSA with ideal biasing is unrivaled, but there is no way that we're aware of to calculate those ideal biasing parameters without, as we did in this presentation, knowing what the true probability is and then trying them all. And the dynamic weighting method used in algorithms like the SWSSA does not necessarily resolve this issue. We see slightly increased robustness, but we still have a narrow range of the possible amount of biasing that really actually converges. And the guided WSSA, along with a motivated method of resolving negatives, would work consistently as the SSA, it would seem, but is too slow. It takes many runs to get to the value, and each run is quite a lot more time complex than these other algorithms. That concludes our presentation. I'd like to thank our collaborators at the Genetic Logic Lab at CU Boulder, as well as the Secure, Efficient, and Evolvable Computing Systems Lab at USF, as well, of course, as our collaborators in the Fluent group at the University of Utah, Utah State University, CU Boulder, and University of South Florida. And then with that, I'd like to open up to questions.
Thank you very much, Payton. Questions? I'm going to check on the Slack. No questions. I have a question, Payton. Mm-hmm. Have you thought about maybe going into parallel technologies for running the guided version of the algorithm to improve its performance?
I have not thought of that. Now, whether my collaborators who are more computing side of things than me have or haven't thought of that, I'm unsure. But I think that's a good idea.
I can add to that, Martin, if you like. So there are some parallel SSA papers out there where people have tried to parallelize it. But the thing with the, so SSA is somewhat embarrassingly parallel, right? Because if you're doing 100 runs, you can do 100 runs on 100 CPUs if you like. That would be the, I mean, people have tried to parallelize individual runs, but because you actually need to do 100 runs that are independent, you could or thousands or whatever, you could just distribute them over multiple cores. But that speed up would not only improve the guided WSSA, but it also improve SSA. So that's the issue, is that, you know, SSA is kind of the prime one, and the whole point here was to see whether you can do better and converge faster with these weighted methods.
Thank you. We have another question in the chat. Jeremy, he's asking, have you documented the 3 original paper examples modifications to the plain algorithm anywhere?
So we, of course, are under pretty harsh time constraints in this presentation, as well as in the abstract for the presentation, which has to be kept to 2 pages, because these algorithms normally take a page or 2 to sort of summarize. But certainly you can find the original papers for each of these and in detail see their derivation of the algorithm.
Okay, I don't know if we have any more questions. If not,
and I would suggest, I apologize, I would suggest doing that as well, because the sort of important sampling method that I present here is simplified for time quite a lot. So seeing exactly how they pull it off, I think is worthwhile.
We have time for one more question. Jen is asking, interesting talk. Do you think similar trend of the 3 methods can be observed on other examples than the futile cycle example? What if an example includes types of reactions such as degradation?
Yeah, so we have implemented, I believe, 7 different models, including a simple genetic circuit, some large chemical reaction networks, some simple chemical reaction networks, a very simple one that was literally just a conformational change between 2 states. And so far, we're seeing the same trends on those more complicated and less complicated examples. But of course, again, in the interest of time, we weren't able to delve into 7 different examples for this talk. But we do think those trends will continue to exist.
Okay, thank you very much, Payton, for the talk.
Yeah, thank you.
Now we're moving to our next talk, which will be given by Gonzalo Vidal. The talk title is LOICA, Logical Operators for Integrated Cell Algorithms. Gonzalo is currently a PhD student in Biological and Medical Engineering at the Institute for Biological and Medical Engineering at Pontificia Universidad Católica de Chile. Gonzalo, please, whenever you're ready.
Okay.
I think there we go.
Okay. Let me do this again. Okay. Okay, I'm ready. So, hi, I'm Gonzalo Vidal, and I will present you LOICA, Logical Operators for Integrated Cell Algorithms, a software tool for synthetic biology developed at Rudge. The design, build, test, learn cycle is central to engineering disciplines like synthetic biology. Mathematical and computational modeling is key to the design stage. Furthermore, there is a need to connect the stages in order to facilitate the flow through the cycle, improving the iteration time. Our aim is to program biology using DNA in order to engineer life. To this end, a programming language could be a good idea to express the designs that will be compiled into DNA on the build stage. We would like to transit from an electrical engineering to a software engineering abstraction level. LOICA is a Python package for modeling and designing genetic circuit dynamics based on a simple object-oriented design abstraction. It uses classes to represent different biological and experimental components, which generates models through their interactions. Python was chosen because it is an open source. It is open source and it has a very active community. It is versatile, readable, easy to use, and fast to develop. is usually taught at Introduction to Programming courses and is one of the most popular programming languages. LOICA, General Mathematical Model, is composed by an operator, gene product, and metabolism. The output is always a gene product, and the input can be a gene product or a supplement. Here, the dynamics of the output are determined by the production term phi, which is a vector of transfer functions mapping input concentrations to output synthesis or gene expression rates. The degradation term gamma is a diagonal matrix of degradation rates of the respective outputs, and the dilution term mu of t is the instantaneous growth rate. The operator is an abstraction of a DNA fragment that encodes a regulatory elements of a transcriptional unit. It is related to the promoter, upstream element, promoter, and ribosome binding site sequences. It can be a NOT operator, a NOR operator, or a node operator with any gene expression function. The gene product is an abstraction of a DNA fragment that encodes for a protein or RNA expressed by the transcriptional unit. It is related to the coding sequence. The regulator is a gene product that regulates gene expression. It is a common operator input, and it could be, for example, a transcription factor like TET-R. The reporter is a gene product that is measurable and emits a signal or emit a signal. It is a common operator output, and it could be, for example, the green fluorescent protein, GFP. The genetic network is the main actuator and encapsulates and coordinates operator and gene product. So here, the operator and regulator can be providing dynamics, and then through the reporter, we can visualize these dynamics of, in this example, a rep oscillator. So this is a plain oscillator. I did not integrate the context of the genetic network. And the genetic network can be, for example, this rep oscillator or any plasmid that you want, if it's stated as transcriptional unit or in the small format. The metabolism provides a cellular context for gene expression, incorporating biomass and growth rate. The growth rate can be a synthetic profile following the Gompers model, or a characterized profile from OD measurements. Here we can see an experimental, from experimental data, we can characterize the growth, and on the blue one, there is the growth characterized using the inverse method that we developed and it will be published soon. Sample encapsulates the genetic network, metabolism, and supplements. Incorporates environment information such as the interaction of the supplements that could be homoserine lactones, for example, or other small molecules. It incorporates information about the strain and the media. It can be, for example, one well in an 96 well plate or a single cell. Assay finally encapsulates a set of samples. It can be, for example, an experiment or the whole 96 well plate. It runs and stores sample temporal dynamics, generates synthetic data, and finally, it can upload data to Flapjack, a data management and analysis tool for genetic circuit characterization. LOICA is two-way communicated with Flapjack. This facilitates the creation of synthetic data and the characterization of operators from experimental data. LOICA, assay, sample, and supplement correspond to the same data model object on Flapjack. Reporter is communicated with signal to produce measurements, and genetic network is communicated with Flapjack vector, which in turn is connected to a small component through a SynBioHub URI. On the mathematical model of a NOT operator, A is the basal gene expression, B is the regulated gene expression, K is the input concentration for half expression, and N is the cooperativity degree of the input. On the code example, we can see an instance of a NOT operator that has that R as input and Ci as output, as well as the other variables stated. To build a repository in LOICA, first we create a genetic network. Then we create and add regulators, in this case like I, that R, and Ci. We can also, and we need reporters to see these measurements. So we add 3 reporters, CFP, YFP, and RFP. Then we add them to the repository. We need 3 NOT gates to make the ring oscillator, and these are the functional part. So first we have a NOT gate that has as input Ci and outputs like I, then a NOT gate that has as input like I and outputs that R, and finally we have a NOT gate that has as input that R and output Ci, closing this ring oscillator. We add these operators to the repository as well. So with that, we build here this part of this diagram, and now we need to add the reporters in order to get the measurements. So here we add extra operators. Here one that has as input Ci and outputs CFP, another that has like I as input and outputs YFP, and finally one that has as input R and outputs RFP. We add that to the repository as well, and then we set the context for this genetic network. So we define the metabolism, in this case, a simulated metabolism. Then the sample encapsulates the genetic network, the metabolism, and provides information about the media and strain. The assay encapsulates the sample and defines the number of measurements and the interval of them. It also provides information about the name, the description, and the biomass signal. After run the assay, we can easily plot the data in Python to visualize the system dynamics. Here we can see that we move from the continuous oscillations, and we can appreciate how the biomass modulates these dynamics of on the repository. Moreover, with one extra line of code, we can upload the data to Flapjack and access to extra query analysis and plotting features through the graphical user interface. So here we can also select and plot just one vector. For example, we can select different strains if we have that on the assay, and provide extra analysis tool such as characterization and more plot options, easy to access. Then another example, here on the mathematical model of a NOR operator, we have a simple tandem promoter equation that is basically a weighted sum of the transfer functions. Alpha sub 0 is the overall basal expression. Alpha sub 1 modulates the transfer function of the input 1. Alpha sub 2 modulates the transfer function of the input 2. And alpha sub 3 modulates the interaction between promoters. This is the most simple tandem promoter model. And on the code, we can make the same equation. And here we put as input AHL1 and AHL2, and as output a synthetic fluorescent protein 1. We set the alpha to be 0.001, so a really low basal expression, 1, 1, and 0. So it's basically just the sum of the individual transfer functions. And both promoters have the same characteristics here in this example. So building a NOR gate in LOICA is just easy as state the NOR gate as a genetic network connected to a vector ID, which is also connected to a SynBioHub URI. We create the reporter, in this case CFP, and the supplements AHL1 and AHL2. Then we instantiate the NOR operator that has as input the AHL1 and AHL2 and output the SFP1 with the same characteristic as the one that I explained before. On the diagram, we can see visually the supplements AHL1 and AHL2 as inputs of the NOR operator that outputs the CFP reporter. Here also we have a simulated metabolism, and one of the features that why it's useful to have this on Python is because we can easily make a concentration gradient in a log space, for example, here. And we can add the supplements at the concentration stated on the for loops. Then we add these samples with different concentration of the two supplements. And as well, we define the number of measurement as well as the interval. We provide a name, description, and biomass signal ID. We run the assay and upload it to Flapjack in order to access to heat map visualization. With LOICA, we are one step closer to program biology using DNA. And on future work, we would like to provide flexibility to use other BioCAD tools, such as iBiosim, generate human machine readable protocols using OpenTrons, closing or restricting the connection between the design and the build stages. We want to expand to single cell and spatial temporal systems. And for that, we also are keen to see stochastic simulation algorithms. We want to include Reducer as a gem product. So this will be one enzyme, for example, and provide more complex models for tandem promoters, such as the road blocking model to the operators. With this, we aim to complete and automate the DVTL cycle in synthetic biology. And we propose here an open source workflow that integrates LOICA, Flapjack, SynBioHub, as well as other tools via SVOL3. I would like to thank the Rudge Lab, Guillermo, Yáñez, Vidal, and Gabriel Miranda. And here is the GitHub repository and the funding agencies. Thanks.
Thank you very much, Gonzalo, for the great talk. Do we have any questions? I'm going to check the Slack channel. No questions there. I have a question, Gonzalo. Have you thought of also integrating LOICA to Cell Modeler?
Yes, that is one of the developments that we want to do, because with LOICA, we can simulate the one cell, for example, dynamics, and then that will be integrated in one object of Cell Modeler. And with that, we will have the connection of this time dynamics to the spatial dynamics provided by Cell Modeler individual-based simulation. So sure, we will do that in future.
Any other questions? I think that's it. Thank you very much, Gonzalo, for a great talk.
Okay, thank you.
So our next talk is called Biophysical Technology Mapping of Genetic Circuits. The talk will be given by Nicolai Engelmann. Nicolai studied electrical engineering and computer science with a focus on communication systems and technology at Technical University Darmstadt. Having come into contact with synthetic biology during his master's thesis, he now works as a PhD student on non-Markovian models of gene regulation and genetic circuits at BCS Lab, Technical University Darmstadt. Nicolai, please, whenever you're ready.
Thanks, Martin, for the introduction. I will just share my screen. Okay, can you see the presentation? Okay.
Yep, it's fine. Yes, thank you.
Nice, thanks. Okay, so I will tell you something about, about a work I did with some colleagues. about thermodynamic circuit modeling and an optimized technology mapping for that purpose, which we simply summarized as biophysical technology mapping of genetic circuits. So for the motivation, I mean, there's this assumption which may be shared by some of you in the audience that computer-aided design of genetic circuits is superior in the long run, because when systems become more complex, computers will be able to process all the information more efficiently than a human can. It's a process which has already happened in EDA before. And currently in genetic design, we have the problem to create functional circuits and functional designs in general. So robustness and predictability is now an issue. And one of the common causes of failures of these things are context effects. So to bring these two things together, we can say that in an automated, robust design process, context effects must be considered, and the design process must be fast enough to allow for this detail. So to give you a short introduction to GDA in general, so usually a GDA toolchain is a function from a behavioral specification to a DNA sequence. So it mainly comprises of 4 segments. So we read in the specification, then there are the 2 core phases, which are logic synthesis and technology mapping for a logic circuit. And then we have some post-processing and throw out a DNA sequence, which should implement the behavior we have specified. And today we want to concentrate on the technology mapping aspect. So what we have there usually is an abstract logic circuit, which we obtain from the logic synthesis part, which basically satisfies in form of a logic circuit the desired logic function. And we now need to assign real technology to it. And this is done in technology mapping. So the single gates must be implemented in DNA. And this technology mapping basically is built upon 2 modules. This is on the one hand, the part specification of the real technology. And then there's some optimization procedure, which iteratively tries to find the perfect technology assignment for the goal to achieve with the circuit. So, and these 2 aspects are currently usually, or these 2 modules are currently usually built like this. So in the specification, we have usually it's phenomenological and data driven, which is not bad per se. But the characterization is pretty simple. So usually we have just transfer functions of gates and in a steady state manner, where we just have an input-output mapping of concentrations of transcription factors or promoter activity or so. And for the optimization, we have usually borrowed stuff from EDA, which tends to be very scalable, like simulated annealing, which can deal with millions of gates in an EDA design process, which is not practical in genetic circuits right now. So it is topology agnostic, which it does not have to be because the circuits are so small. And they're very complicated to evaluate. So the simple characterization somehow contradicts that as well. So to get to this specification part first, like I said, in the classical case, we equal gates until transfer functions if you want. So we abandon all the knowledge we have on the subgate level. Context effects, however, happen on the subgate level, and they alter the circuit's response, like crosstalk and titration effects with the host. And we can calculate this on the subgate level. One of the more economical models for this, which model these aspects, is a thermodynamic circuit model. So this thermodynamic circuit model allows us basically to extend or expand a given logic circuit by incorporating host gate crosstalk, gate crosstalk, and gate host titration effects. So what are those? Host gate crosstalk is basically crosstalk from naturally occurring transcription factors in a host, which may interfere with the signals in the circuit. Gate crosstalk is the same, which is basically just coming from another gate through imperfect orthogonality. And gate host titration comes from the fact that in a host there might be competing binding sites for transcription factors present, which drain those from the circuit basically. So, and the question how this works is dealt with in many papers recently. And it's basically we could say it's called an equilibrium thermodynamic gene expression model. And the idea of this is we model basically a DNA compound where polymerases and transcription factors can bind as a canonical system, which is in an equilibrium with the heat bath. And so the idea comes basically, or it is just realizable because there is this timescale separation between the signals in a circuit we are interested in, or what we can measure if you want, which is a protein expression level, and the binding and unbinding of polymerases and transcription factors. So the one thing happens in minutes and hours and the others in seconds. So we can, if we take a look at binding and unbinding of polymerases and transcription factors, always look at the equilibrium. And such a canonical system which is in an equilibrium with a heat bath can be calculated, can be described by a Boltzmann statistic. And this Boltzmann statistic is dependent on the energy level of the system. And using this, basically, we only need to find, we can assign energy levels to each permutation of binding and unbinding of polymerases and transcription factors, and isolate basically the cases which are interesting for us. So usually we're interested when we have a promoter and a gene downstream of that promoter, which we want to express. We usually have basically we are interested in the cases where the polymerase is bound to that promoter. And this basically leads us to we can describe this with the Boltzmann statistic, and this basically leads us to the second aspect of this or the second assumption of this thermodynamic gene expression model, which is that the probability that we encounter a polymerase bound at this promoter of interest, so that the gene is expressed, is then proportional to the number of proteins we can measure in the end. So the train of thought here is basically, if a polymerase is double as likely to bind to a promoter, we can think of that over time it has double the amount of being bound to that promoter, and thus we can see the double amount of proteins in the end. So this is basically the idea. So, and the transcription and translation basically is a black box in this in this idea. We just model this as a constant, if you want, so as a coefficient resolved by this proportionality. So, and to give you an example how this would look in a gate, we have derived a NOT and NOR gate here in this case. So we use basically also this NOR logic semantic, which is also used in cello. Without modeling the titration, we can model the crosstalk there easily. So let's say we have a gate here which has n promoters as input, so there are n input signals, and there is a certain transcription factor carrying the signal in this gate. So if all gates are not NOR gates, they are all repressors. Yeah, so all these transcription factors are repressing the promoters, reducing the promoter activity. And usually we have only these n transcription factors which we want to bind to the specific promoters. However, usually there are others present which can also bind, or they have a crosstalk among each other. They're not perfectly orthogonal, and some stuff may come from the host, some may come from another gate. And we can model this by these additional crosstalk signals, basically. Yeah? So if we have k transcription factors present, then we have k minus n crosstalk signals, which also may be not of any importance, but we can model them. And we have basically these circuit signals which are wanted. And then we can calculate the fold change for this gate, for this transcription factor which is expressed by the gene in this gate. And we can see that this is a weighted sum of regulation factors of the single promoters. Yeah, and in the single promoters, we basically, these are the regulation factors here. In the single promoters, there are all these transcription factors present, all these K ones, yeah? But they do not have to have an influence. So if we have perfect orthogonality, we can just set the energy terms to specific values and they will just disappear. But we can model an arbitrary binding affinity of these several present transcription factors to the single promoters. And we have then also these weights, basically. So the single promoters are also weighted differently with respect to the energy level of binding of the polymerase to this promoter, which is also nice. So now we have a gate. Yeah? We can say that if we want to have a circuit, we have this gate function, which comprises of a fold change and a proportionality coefficient. Like I said before, this is one of the main assumptions. And in this case, this would be the unregulated gene expression. So if we have no transcription factors present, how much protein would we observe in the end of these input transcription factors to this gate? So this multiplied by the fold change gives us the number of this expressed transcription factor. If it's a transcription factor, it can also be a reporter or whatever. And we have now basically per gate, we obtain another unknown, yeah, which is a molecule which we need to count. And if all the boundary conditions are known, so all the inputs are known to the circuit and also what comes from the host, then we can calculate basically this as an equation system of M unknowns and M equations, so it is solvable, with which we can calculate a whole circuit. So this is how we do this. So a consequence of this is basically we have now a parts library which is built on a smaller scale than a gate library, which also contains binding energies between polymerases, transcription factors, and promoters in the host genome, for example. And we have basically, if we want now to model non-imperfect orthogonality, we also need to consider cycles in the circuit. So the circuit will not be combinatorial in nature anymore. So you cannot forward propagate your signals with a finite composition of functions. You need to do a fixed point calculation by an iterative algorithm. So in this case, you could basically use a quasi-Newton. And from our estimations right now, from what we already know, on average, we will have around a 30-fold runtime increase through this. If we want to calculate a circuit which has been formally calculated by in a standard way by Hill functions, basically by compositions of Hill functions, and you want now to do the thermodynamic calculations, you will on average have 30-fold the runtime. And the scoring has been done with Jello score, so the on-off separation basically. So having this 30-fold increase now, this is now a more complex mapping. So our goal is simultaneously to reduce the number of circuit evaluations which are necessary. And like I said, there is this topology unawareness in this in the classical mapping algorithms, basically, in this case, simulated annealing. So it has a good runtime and it has a good mean solution quality, but it suffers from stochasticity. So basically, if you would obtain a result from this designer, you would ask yourself how good it is really, and probably not enter lab phase with that. So we introduced basically this branch and bound based mapping in addition, which can guarantee an optimal result. So we first gather the information we have from the circuit topology, the logic states of the abstract logic circuit coming from logic synthesis, and the information we get in the parts library. And then we do basically a standard branch and bound on the reverse topology of the circuit. So you need, for branch and bound, you need a DAG, and the DAG is already given by the circuit if you want. So we basically, we start from the last gate and then iterate until we have the full circuit basically mapped. And within these steps, when we have not a fully mapped circuit, we need an optimistic score, an optimistic estimation for the score. So this is basically our bounding. And if we can, by this optimistic estimation, find inferior solutions, we can prune them without any problem. So this is basically the core of this branch and bound. We have an optimistic estimation of a global solution which is reachable with a partial solution. So it must be, if it is truly optimistic, we never prune solutions which could lead to an optimal solution. So we never abandon solutions which might be really good. And on the other hand, we must estimate realistically, so that we do not basically in the end do an exhaustive search. So we must also be able to prune these subtrees of solutions. So, and this estimator is highly problem and model specific, and it works not only with the thermodynamic model, but it can also be applied to the classical function model. So what I want to tell you now is how we basically do this bounding. We can recap here. We have a partial solution, and its quality is basically determined by an embedding in an optimistic global solution. And the mapping is extended in reverse topology order. So we start with the last gate and end with the first gate, if there's even the first gate. So we need an estimation for the input intervals at the specific subcircuit we are looking at. So this partial solution, if we want to have an optimum estimate of what it gives as a score, we need to give it the best possible inputs so that it can lead to a highest score, so that the real score is in any case smaller than the one we are estimating. So we're basically trying to find this optimistic input, and this can be found by taking extreme values of all the technology which is still available in the library, so that any real technology assigned to the circuit which is still unassigned can only lead to a worse result. So yeah. On the same hand, we can rule out certain Boolean input combinations because they're sometimes just not possible by logic. So if we take these extreme values, okay, they always correspond to a 1 or a 0 for the specific wire. And we can basically see in the circuit topology already that certain combinations are impossible. Like in this circuit, we have A and M1. A is also connected to this NOR gate, and A is on this upper wire here. So a combination A equals to 1 and M1 equals to 1 is simply not possible. So we do not need to take these values in account, speeding up the process at the same time, and allowing the optimistic bound to be a real upper bound. So we have evaluated this, and for this we have mapped 66 circuits with 2 to 8 gates. And those are built from the 33 Boolean functions introduced in the Cello paper. And we have mapped 2 topologies for each one. This is on the one hand, it's the topology from Cello, and on the other hand, the optimum structural variant we have obtained in our work in the last IWBDA. And as an objective function, we use the Cello score. So, and there are 2 branch and bound modes we use. This is on the one hand the optimal mode, which guarantees the optimum, which is basically as good as exhaustive. And then a heuristic mode, where we basically can also prune subtrees which are not perfectly optimal. So in the optimal branch and bound, we can see that we outperform exhaustive search by 24-fold. So we're 24 as fast as our exhaustive search by having also a guaranteed optimum, so with the same result. And with a heuristic branch and bound, we outperform simulated annealing 70.5-fold, while simultaneously obtaining better solutions, which means that in 20% more cases, we get the real optimum compared to SA. And on the same time, the difference between our near optimal solution and the true optimal solution is decreased dramatically by 600,000-fold. So usually if you use the heuristic branch and bound, you always get a solution which is pretty, or almost always get a solution which is pretty good. We can see it down here in numbers. You have to notice that this is in logarithmic scale here. So if you compare exhaustive with the heuristic, that's really crazy. So you can multiply 70.5 times 24 if you want. Then you get your fold change. So, and yeah, to conclude this, we have developed so we have incorporated some context effects in technology mapping using a thermodynamic gene expression model for the circuit. And in the same time, we have sped up technology mapping by using a topology-aware branch and bound method. And this will also be integrated in our design toolchain, in our robust design toolchain Arctic, where there's the link down below. I will post it in the chat after the talk, where you could check out it regularly. Usually the changes and improvements we make in our research are then worked in this tool. So if you want to use it to make robust circuits, you can do. Right now, it can deal with structural variance and cell-to-cell variability, which is already pretty cool. Thank you for listening. And I also want to thank my colleagues, Tobias and Erik, especially for their work on the branch and bound, and Christian Hochberger and Heinz Koeppl for the supervision. And I would be now open for questions. Thank you.
Thank you very much, Nicolai. Do we have questions? I have one.
Yeah.
Oh, sorry. I'll ask mine after Hatem's, maybe.
Okay.
Just waiting on the question. Oh, okay. I'll go ahead. You talked about simulated annealing as the heuristic method for improving this branch and bound. Have you thought about other heuristics?
Yeah, it's so simulated annealing is basically it's the state of the art right now. So you usually have let me just go there. I'm sorry, it was not clear. In the state of the art, like in the cello framework, for example, you have simulated annealing optimization to do your technology mapping. And the problem here is that the simulated annealing is just borrowed from EDA. So it is built for circuits which are on a completely different scale in terms of size and complexity than those are in genetic logic circuits right now. So we basically introduced a completely different approach, which is branch and bound, which goes through the topology of the circuit. And the reason why this works better is, in our perception, that the performance of a genetic circuit, or maybe also of an electrical circuit, but we don't know, depends strongly on the performance of the last gates, especially when you want to score them by an on-off separation. So think about a gate as a last gate where you have a high fold change between on and off, and another one which has a very small fold change between on and off. You will never choose this one with the small fold change, because whatever you do before, you will anyway have this small fold change in the end. So the idea is basically to assign the best gates to the final logic gates in the cascade, so that you already start with your best estimates about your fold change, basically, and then fill up the rest so that it works. So this is basically the idea, and because of that, it is a lot faster than simulated annealing in this case.
Amazing, thank you. We have a couple more questions. Hatem, how thermodynamic modeling differ from kinetic modeling? Would it not be possible for kinetic modeling to capture context effects as well?
Yeah, this is possible as well. You can, of course, also write down the kinetics between the promoters and the non-cognate transcription factors. But what is nice about this thermodynamic model is that it gives a simple way to describe such effects. So you basically have your canonical physical system, and you just need to know energy levels to perfectly define what is happening between your components. And it is very economical in terms of parameterization. So if you could basically just measure these energy levels between different pairs of TFs and promoters and so on, and then you would have all the information you needed, which is pretty nice to model a complete circuit out of it, if you want to just incorporate these effects.
And I have one more question from Aaron. He's saying, did you recreate the annealing in cello, or did you use any other formula to calculate the acceptance probability?
We have our own custom formula because the simulated annealing in cello is, I don't want to hurt anyone here, but it's really not optimal. And therefore we basically at first, our first step was to optimize simulated annealing on the cello library, and then use this for comparison. So the annealing schedule is different, and the way basically, yeah, that's different.
Okay, I don't think we have more questions. I'm gonna just quickly check on the Slack channel. Nope.
Okay.
So thank you very much, Nicolai, for a very interesting talk.
Thank you as well, Marty.
So we're going to move on to our last talk, which is automated translation of logical models to SystemVerilog enables simulation speedup. So this talk will be given by Erik Lee. Erik is a senior undergraduate student in the Melody Lab at the University of Pittsburgh. So Erik, please, whenever you're ready.
Thank you for the introduction. I guess I'll get right into it. Okay, so like mentioned today, I'll be talking about the automated translation of logical models to SystemVerilog, which will enable system speedup. And what this means will become more clear as I go through the slides. So before anything else, we should first talk about what we even mean when we say model. So in this context, a model refers to a computational model that generally models some cell function. So the elements represent genes, proteins, small molecules, and biological processes. And the edges between these elements represent physical or causal influences. And these edges also have polarity, so it will show which elements will regulate what other elements. And we use models because they require less time than just using in vitro or in vivo methods for hypothesis generation. And more specifically, logical models do not depend on reaction rates, which allows multiple benefits. One being that it reduces time for model assembly and model execution, as well as allows us to represent other different molecule types, either because they do not have as defined literature, so they do not have a certain reaction rate associated, or they can just be more abstract concepts such as apoptosis or proliferation in the example model. And when we actually represent these models as executable simulations, we take a model, for example, this one right here, and translate those into update rules. So for example, here it shows that C at the next time step is equal to A or not B. And also these these update rules are the relationships between the regulators. And when we actually go to simulate that, we can see that it can accurately predict dynamic model behavior. So now we have a great tool in models. An issue, however, is that for any sufficiently large model, or sufficiently large system, we will also have a large model where each of these models can have hundreds or thousands of elements, and each of these elements can have multiple interactions. For example, AKT, you can see how many interactions it has. And also further compounding the issue, feedback and feed-forward loops can also add to the complexity. And so if we go to simulate these models, we can see that software simulations could be too slow, or we might not be able to do everything we want with it. For example, if we wanted to run multiple models with different initialization values in parallel. And this is possible with software simulators, but it could require expensive hardware, expensive resources, or again, would take too long. And so one way we can deal with this is through the use of FPGAs, which for those who don't know, are integrated circuits that are reconfigurable. And they offer many benefits over software simulations, but they provide spatial parallelism, instruction efficiency, and reprogrammability. So we can just load a new model and the FPGA will automatically convert that into a physical hardware. And these are written in are encoded in HDLs, and the HDL that we chose to use in this project is SystemVerilog. As one benefit of SystemVerilog is that it can allow for the creation of configurable and scalable models better than other HDLs. And so we have an HDL which can simulate models through hardware, and we have models which are a great benefit. So where do we go from here? So the problem now, however, is that model building is quite time intensive and would require both domain knowledge in the model that you're making as well as in FPGAs. And currently most published models are not in HDLs. They're either just described, or they are made to be specifically run in software simulators. And if we wanted to convert these logical models, we had to do so manually. And if they had thousands of lines of code, that would take quite a while. And so here in this work, we propose a tool that allows for the translation of several different logical models in different formats into an HDL, SystemVerilog, so that we can run them on hardware simulator. This is the general workflow of the system, where at the top, at the very beginning, we take different sort of logical model formats. I'll describe each in detail a little later, but we take biorecipes, which is a format that we use in our lab, and we also use Boolean expressions, which we can download from the database Cell Collective. We feed these into our translator along with some initialization values of just what elements should be initialized at the beginning of the simulation. And that will produce certain components of the hardware simulator, the logical model and the initials. So the initial will just hold how many elements there are, the random seed if we wanted to do, if we wanted to incorporate RNG into our models, and the logical model, which holds all the update rules for the elements. This combined with the other elements of the hardware simulator will, which allow for, which tell the simulator when exactly to update the elements and how to update the elements. Those together can be put into ModelSim, which is a hardware emulator which produce simulation results. And not in this work, but obviously with all these, they can then just be uploaded into physical FPGA. So to delve a little deeper, this is obviously the translation of the update rules into the HDL. So again, we have these update rules from a executable model, and these are converted into their SystemVerilog equivalents, which then will, when uploaded to an FPGA, will be physical circuits. So one of the formats that we chose to use is biorecipes, and this offers multiple benefits. One is that it's represented in a tabular format, and each line in that table is for one element. And it's quite abstracted, so experts who are not familiar with binary logic and things like that can also use it as well. And also executable models can be directly generated from this. And just a little more about the actual notation. If you want to AND two regulators, it would be in parentheses, and if not, it would be an OR if it was no parentheses. And also not in this, not using this work, but potentially future works. It also has different notations for discrete models. So for example, capsase 367, this shows that it only regulates when, or only activates when it's at its highest state, or you can assign different weights to different regulators, so on and so forth. In addition, we also use Cell Collective, which is a database full of different logical models, over 200, and a Cell Collective provides additional information on each model, and also if there's any associated papers with those models. And so from Cell Collective, we can download these models as Boolean expressions, as you can see here below, and the translation of these would be quite straightforward. And for the hardware simulator that we use, we use a hardware simulator that was published by the lab in a previous work. And as you can see, there are many different update schemes, either simultaneous or random or sequential, so on and so forth. And as you can see, the hardware simulation over the software equivalents produces a varied speed up, but generally it's about an increase of 2 orders of magnitude. And for this work, just for demonstration purposes, we choose to use only one of the update schemes. We chose simultaneous, where we just update all the elements all at the same time. With that being said, here are the models that we simulate. And we chose these for multiple of reasons, one being that they're all large, complex models, and also they offer different sort of signaling cascades that differ from one another. And so we simulate these models just to test the ease of use and accuracy of the translator. So, and all of these models highlight a specific signaling cascade that is highlighted in an associated paper. So here's the first model that we chose to simulate. It's from Biorecipes, and there are 47 elements within the model. And as you can see, the signaling cascade that's highlighted is a incoherent feed-forward loop between apoptosis and BID. And just like in the software simulations, we see that the hardware simulation also predicts the activation of BID. This is the second model that we chose to simulate. This had 247 elements from Cell Collective, and it shows the indirect that EGF indirectly activates AKT. And we see a different sort of signaling cascade. And finally, here's the last model that we chose to simulate from Cell Collective. It had 139 elements and shows a different activation of a PP2A through other elements besides EGFR. So here's all those simulations, and we noticed that regardless of the model size, they were all within a couple of milliseconds, which was a little slower than some of just the hardware, but still much faster than any. The translation of these models took a couple milliseconds, which was took up the majority of it rather than the hardware simulation part of it, and was faster still than any software simulator. So in conclusion, these models allow us to explore signaling cascades much faster than if we would just relied alone on in vitro or in vivo methods. and logical models are often complex, so software simulations might not allow us to do everything that we'd want with it. And so we rely on FPGAs, which can simulate these models much more efficiently and accurately. And our tool here allows the conversion of Boolean models into HDL code so that we can take previous models without much effort and simulate them through hardware. Some future directions that we want to take this is that we want to translate other types of formats, including truth tables or adjacency matrices. And we'd also want to include discrete models rather than just logical models, which have 3 or more activity levels rather than just 2. I'd like to thank all the other members of the Melody Lab for their collaboration throughout the project. And thank you for listening.
Thank you, Erik, for the great talk. Are there any questions for Erik, please? Jen is asking a question. Does your translator convert to combinatorial or sequential circuit modules in SystemVerilog? If it is the latter, do you make assumptions about the global clock for this conversion, as biological circuit models are naturally asynchronous?
Right. So in this, we chose to simulate these through, for this work, we chose to simulate through simultaneous, but the hardware simulator offers other sorts of simulation schemes like asynchronous. But for here, we are making assumptions about the clock. Yeah.
I have a question about the future directions you mentioned. Mm-hmm. You put the adjacency matrices. Those are for to describe the circuits in another format. What is it specifically meaning?
To be honest, I actually don't have much knowledge in that area. It's actually something that my collaborators are working on, but so I'm not too sure.
Okay, more questions? No questions on the Slack channel either. So thank you, Erik. Well, this concludes our session.